In this section, we're going to go into stage 2. So just a reminder of the 4 stages that we're looking at. The first one was the lymphocyte development and clonal deletion. Second one, which you're looking at here, is the presentation of antigen and clonal selection. The third, we're going to be looking at the challenging of B and T lymphocytes by antigens. And then finally, at the end, we'll look at the T lymphocyte and the B lymphocyte responses. So here, looking at presentation of antigens, the first thing is the entrance of antigen. So in order for something to be perceived as an antigen or an immunogen, it has to meet specific requirements in terms of foreignness, shape, size, and accessibility. Foreignness, we're talking about a quality of not being a normal constituent of the host. Whether it's a whole microbe or a part thereof, cells or substances that come from other humans or animals or plants or various molecules possess this quality of foreignness, and so they are potentially antigen. Molecules of complex composition, like proteins and protein-containing compounds, prove to be more immunogenic than repetitious polymers, which are made of a single type of unit. Most materials that serve as good immunogens fall into certain chemical categories, and those are proteins and polypeptides, which would be your enzymes, cell surface structures, exotoxins, etc. Lipoproteins, which are found in cell membranes, glycoproteins, blood cell markers, nucleoproteins, like DNA, which is complexed to proteins, and polysaccharides, so certain bacterial capsules, as well as lipopolysaccharides. Now, good immunogens. So an antigen can be of varying degrees of immunogenic. Characteristics of a good immunogen, which means it provokes a strong response, are their chemical composition, their context, meaning what kinds of cytokines are present, and their size. So generally, you could say large antigens are better than small antigens. However, large size alone is not going to be sufficient for immunogenicity. Glycogen, for example, a polymer of glucose, has a highly repetitious structure. So it has a molecular weight over 100,000 Daltons, but it is not normally antigenic. On the other hand, insulin, which has a molecular weight of 6,000 Daltons, can be antigenic. So this aspect of size relating to the ability to stimulate immune wakefulness. The smaller epitope to which the awakened immune cells bind, initiating the cascade of events to follow, is the one that's of relevance. Here you see a comparison of good immunogens and poor immunogens. Here, the good ones, they're large, they're complex. And then when you're looking at the bottom, at the smaller one, the poor ones, these are less likely to be good immunogens. They're repetitive, they're not complex. Everything here you look at is much more complex in structure. The term haptens. Haptens are small foreign molecules that are too small by themselves to stimulate the immune response. But if this, I'm going to call it an incomplete antigen. So if this incomplete antigen is linked to a larger carrier molecule, then that final product, the combined molecule, can become immunogenic. So the carrier group contributes to the size of the complex, and it enhances the proper spatial orientation for contact, while the hapten is the one that serves as the epitope. So haptens include molecules like drugs, metals, and ordinarily innocuous household, industrial, and environmental chemicals. Many of them inappropriately develop immunogenicity in the body by combining with larger carrier molecules like serum proteins, for example. So here you see a poor immunogen that has bound to a carrier molecule, and then it becomes a good immunogen. Alloantigens. Because each human being is genetically and biochemically unique, unless we're talking about identical twins, The proteins and other molecules of one person can be immunogenic to the other. Alloantigens are cell surface markers and molecules that occur in some members of the same species, but not in others. So they are the basis for an individual's blood group and for major histocompatibility complex profile. They're responsible for incompatibilities that can occur in blood transfusion or in organ grafting. Another term, superantigens. So superantigens are a group of immunogens. Bacterial toxins are found to be a part of this, and they are potent stimuli for T cells. So if they are present in an infection, it activates T cells at a rate that is 100 times greater than ordinary antigens. The result can be an overwhelming release of cytokines and cell death. Such deaths as toxic shock syndrome and certain autoimmune diseases are associated with this class of antigens. When you have this overwhelming release of cytokines, it's called a cytokine storm, and it plays a role in several very serious infectious diseases. Cytokine storms have been recently cited related to severe COVID-19 cases, and they also played a role in influenza deaths and in the pandemic form of Black Death. Now, looking at antigen processing and presentation. In most immune reactions, the antigen has to be formally presented to lymphocytes by cells that are called antigen-presenting cells. At least 3 different kinds of cells can serve as antigen-presenting cells: macrophages, B cells, and dendritic cells. Dendritic cells are the most potent and the versatile of the 3 different antigen-presenting cells. What they do is these APCs grab the microbe and they ingest it, they degrade the microbe, and they pass its antigens back out into their membranes, complexed with either MHC1 or MHC2 markers that are already there. Now it's accessible to T lymphocytes for presentation. So here you see an image which is portraying what I was just talking about. You have antigen-presenting cells. They are engulfing a microbe. They take it into intracellular vesicles, break it down into smaller peptides or pieces. These pieces are then transported to the membrane of that antigen-presenting cell. So here you see that in this image. They're being transported and then presented there. They form a complex, in this case with MHC2 receptors. So from this surface location, the antigens are presented to a T helper cell, which is specific for the antigen that is being presented. So this is an example with MHC2, could also be with MHC1. Now, first, the MHC2 antigen on the APC binds to the T cell receptor. After that, a co-receptor on the T cell, CD4, cluster of differentiation 4 typically, it will hook itself to a position on the MHC2 receptor. So there is a double binding combination which happens, and this ensures that there's simultaneous recognition of the antigen as well as the MHC receptor. So the antigen being non-self and the MHC receptor being self. An additional molecule on the surface of the APC, which is called CD80, is also needed for efficient activation. It interacts with the CD28 molecule on the T helper cells. Now, once identification has happened, the APC will activate the T helper cell, also TH. The APC also secretes interleukin-1, IL-1. The TH cell in turn produces interleukin-2, which is a growth factor for the T helper cells and cytotoxic T cells. These T helper cells can then help to activate B cells. So most antigens have to be presented first to T cells, even though they'll eventually activate both the T cell and the B cell systems. But there are some antigens that can trigger a response directly from B lymphocytes without the cooperation of antigen-presenting cells or T helper cells. Those are called T cell independent antigens. They're usually simple molecules like carbohydrates, which have many repeating and identical chemical groups. So examples include lipopolysaccharide from the cell wall of E. coli, Escherichia coli, and polysaccharide from the capsule of Streptococcus pneumoniae, and molecules from rabies and Epstein-Barr virus. Because so few antigens are of this type, most B cell reactions require T helper cells.